Hello, and welcome to this Nature.com custom webcast titled Bringing EDS to Life, Sample Considerations for Biological Element Analysis. My name is Jayshan Karpan, and I will be your moderator. Today's webcast is sponsored by Oxford Instruments. We'll begin the webcast with presentations and then end with a Q&A session. To ask a question, just type it in where it says type your question here and then press submit at any point during the webcast and we will answer them today. And now over to Dr. Louise Hughes of Oxford Instruments to introduce today's webcast.
Thank you for the introduction and welcome to everybody watching. I am Dr. Louise Hughes from Oxford Instruments Nanoanalysis, and I am joined today by Erin Johnson from the William Dunn School of Pathology at the University of Oxford. We will be discussing using EDS in the life sciences, with particular emphasis on the ways in which sample preparation can be used to address different biological questions. We have broken the webinar down into a few sections, and I will start off by giving a short introduction into the production of different signals generated in an electron microscope when an electron beam interacts with a sample, and how EDS can be used to detect one of these signals. We will go on to consider the generation of EDS data, followed by how biological samples can be particularly challenging for this technique. I will then talk through a few examples before handing you over to Erin, who will talk about their current research using EDS on biological specimens. I will then give a brief summary before we move on to the question and answer session. Please feel free to ask questions during the webinar, and we will endeavour to answer them at the end during the question and answers. First, I wanted to talk about signal generation. When the beam of electrons hits a sample, several signals are generated. The beam of electrons penetrates into the sample as demonstrated by the onion diagram on the left. The higher the accelerating voltage, the larger this interaction volume is. There are several signals generated as a result. The diagram on the right shows a basic schematic of an atom and more details of the electron interactions that can occur. You can see on the diagram several electron layers around the atom's nucleus. The number of these layers and the energy of the electrons within these layers is characteristic of each element. In a TEM, the normal bright field image is produced as a result of electrons being transmitted, as shown in green in the diagram on the right, or scattered by elastic and inelastic scattering events, which creates contrast. More electrons are scattered by heavy elements compared to light, which is why in a TEM you need a density difference between your samples and the surrounding substrate, something that has been traditionally increased using heavy metal stains. Scattering is also important for SEM and produces a range of signals including secondary electrons, backscattered electrons and characteristic X-rays. These signals are generated from the full volume of the specimen beam interaction, but you can see from the onion diagram on the left they are only able to escape the sample and be detected from certain depths. One type of electrons detected from biological samples are backscattered electrons, which are main beam electrons that have had their direction of travel changed by passing close to an atomic nucleus, resulting in them being scattered back out of the sample. Backscattered electrons are high energy and thus able to be emitted from deep within the sample. Secondary electrons are generated when electrons orbiting an atom within a sample are sufficiently excited to leave the atom, and this occurs when energy is transferred from a main beam electron. Secondary electrons are relatively low energy electrons and are only emitted from the sample if they are generated fairly close to its surface. The third signal is X-rays, which are produced as a result of the generation of secondary electrons. Once the secondary electron has left its orbit, it leaves a hole in the atom. In order to fill the hole left by the secondary electron, an outer orbiting electron releases energy and moves to fill the gap. This energy is emitted in the form of an X-ray and relates to the energy difference between electron layers. Thus, detecting the energy of the electron allows us to identify the element it originated from, which is what we detect using EDS. EDS, also referred to as EDX, is energy dispersive X-ray spectrometry and refers to the technique of using a detector to measure the X-rays emitted from a sample when exposed to an electron beam, as discussed in the previous slide. In recent years, that has been done using a silicon drift detector. The signals are then processed electronically and displayed as a spectrum using Aztec, our EDS software. The position of peaks in the spectra relate to elements in the sample, and the height of each peak relates to how much of it is present in the sample. Spectra can be produced from a single point or at each pixel position as a beam scans across the sample, as it does when producing an image in a scanning electron microscope. By collecting a spectrum at each pixel, an X-ray map displaying the locations of different elements can be generated, as can be seen in the image at the bottom right of this slide. Here is a closer look at a spectrum generated from the sample shown in the electron microscope image at the top left of the slide, featuring macrophages grown on a plastic cover slip. The macrophages have ingested some chromium particles. When we look at the spectrum for the area covering the field of view shown in the image, you can see a range of peaks and labels. Along the x-axis, we have the X-ray energy shown in keV, and the position of the peaks relates to the characteristic energy of the X-rays collected by the detector. Larger peaks show that more X-rays from that element have been detected, and there are more of those atoms in the sample. Aztec performs an auto-identification of elements and labels them as shown on the spectrum. You can see that sometimes there are several peaks from the same element. That is because you can get a variety of electron interactions in the specimen on different layers, resulting in the release of X-rays with several different energies. But this can only happen with heavier elements. The range of X-rays produced from light elements is restricted, and they are usually clustered at the lower energy range. There are two things to note here. In general, if you want to achieve the higher energy peaks, you may need to use a higher accelerating voltage, at least 2.5 times the voltage that you are hoping to activate. This is generally more of a challenge in SEM, as TEM voltages usually mean this is not an issue. The second point is that many biological elements are light elements and do not have higher energy electron interactions. In those cases, increasing the kV will not help to extract more information. It is important to use an EDS system that is sensitive enough to not only detect light elements, but software that can deconvolute peaks when they overlap each other, such as Aztec. Determining your microscope operating conditions by what elements you expect to see in your samples is extremely helpful when planning your experiments. An important part of EDS is detecting as many of the X-rays as possible. X-rays are emitted in all directions. Smaller detectors will therefore detect a smaller proportion of X-rays. How close the detector is to the sample also plays a role. A smaller detector closer to the sample will count more X-rays than a larger detector a long distance away. We have optimized detector performance by producing very large area detectors, 100 and 170 mm squared, which are located as close to the specimen as possible. Detectors typically are kept very cold and under vacuum. In order to do this, they have a window on the detector, preventing moisture from condensing onto it when you vent the chamber. However, we also have a new type of detector, the Ultim Extreme, which has eliminated this window. The Ultim Extreme detector has a much smaller snout and no window compared to traditional detectors. This means two things. The first is that it can get closer to the sample, as you can see in the diagram on the left. The second is that more low-energy X-rays, those typically produced by light elements such as those present in biological samples, and which can be absorbed by the thin window in traditional detectors, can now reach the silicon drift detector. This means we can also image the sample at lower accelerating voltages than before, which in turn reduces the specimen beam interaction volume and improves resolution in images and in signals. On the right side of this slide you can see a comparative image of a Venus's flytrap leaf gland captured using the Ultim Max 170 and the Ultim Extreme. The top images show X-ray maps over the electron images. The bottom two show the X-ray maps on their own. The data was captured at 6 kV in 15 minutes. As you can see from the images and the spectra, more signal was picked up by the ultimate stream in the form of more counts, and nitrogen in particular, shown as yellow on the images, is much more apparent. It should be noted that this is a bit of an unfair test for the Ultim 170, and the optimized conditions for that involve using higher KVs, but hopefully it demonstrates the advantages of using the Ultim Extreme at lower KV settings. The results presented in the rest of this presentation will be generated from 3 of our detectors. The Ultim 170, shown on the left, is our largest area detector and is ideal for fast analysis of heavy elements where you might require higher accelerating voltages, and is typically used at 10 kV or higher. The Ultim Extreme is our windowless detector for SEM, and I have just shown you some results generated at lower KVs, although you will see that we can generate data at much lower KV than this. Our Ultim Max TLE is our flagship windowless detector for TEM, and is the detector used for all of the TEM data presented later on in this presentation. I have described the technology a lot so far, but this is the type of data shown on this slide that we want to produce, images of biological structures. The image on the left is from TEM data and on the right is SEM data. This is from the same type of sample, an unstained leaf, and you can see cell ultrastructure just using maps of elements without the need for stain. While these images were collected on samples being imaged at room temperature, it is also possible to do this under cryo-EM settings. Hopefully you can start to see how EDS can be used for colour EM imaging, adding compositional information in the form of colour as an additional dimension to your data. We have already discussed the spectra and how they can be collected at each pixel position when the beam scans across the surface. Here is a lily pollen grain prepared for normal SEM imaging and coated in gold to increase the conductivity of the sample. I was interested in producing some elemental maps of the pollen grain, and you can see one of these at the top right. When processing the data at a later date, away from the microscope, I noticed an unusual structure on the surface, which I have circled in yellow. I collected a spectrum from the circled area. You can see the gold peak, but also peaks for iron, magnesium, aluminium and silicon, which were unexpected, and this compositional information indicated that the particles were airborne pollution, a subject we covered in a webinar a few weeks ago. Because the data is already in the image, I could collect spectra after imaging had been completed, and I can revisit this data for further processing at any time. I wanted to talk to you a bit about what we can expect in biological samples. Here is a typical periodic table of elements, most of which can be detected using EDS. When we come to the composition of biological samples, the table looks a lot more like this. You can see immediately that there is a reduced range of naturally occurring elements found in biological samples, and these are mostly the lighter elements on the table. Therefore, it is important that your detector has the sensitivity to detect these if your research is focused on native elements. Of course, the range shown here may be different if different stains have been used for contrast, or if there are externally derived materials, such as implants used in medicine, nanoparticles used for drug delivery, or heavy metal pollutants. All of these elements, except hydrogen, can be detected using EDS. One of the key features that can prove problematic for EDS is the relative abundance of each element and whether it can be detected above background noise. The concentrations of various elements in humans are given in the list towards the bottom of the slide. This is a general list encompassing all tissues. Elements may have different concentrations in different tissues and within tissues, cells and organelles. Research in this area is still very limited, and there is much we don't know about elemental localization at the ultrastructural level. There is still no complete list for elements in a single organism. over 50% of enzymes depend on metal ions to function, and over 3,000 proteins alone require zinc. EDS is a technique that can help us answer some of the questions that have yet to be answered, and the recent improvements in detectors and processing technology have made the analysis of biological samples not only possible, but fast and a routine method that can be incorporated into any microscopy lab. Some of the challenges for EDS using biological samples are listed on this slide, some of which we have already touched upon, and the potential solutions. We have already discussed how biological samples are composed of light elements, and to address this you need sensitive detectors, with windowless detector design being one of the key areas. It's also important that we have low kV capabilities for SEM, especially with samples that are non-conductive. As you can see in the image on the right, you also get non-homogeneous samples. This means that we don't have isolated regions of a sample composed of a single element, which can occur in some materials samples. Biological samples by their nature are non-homogeneous, and this makes mapping really difficult because we've got lots of elements overlapping one another. So it's important to have solutions where you can deconvolute the peak data for accurate mapping, and low kV that improves spatial resolution. One of the questions I frequently encounter is what type of biological sample can we do EDS on? If you can prepare a sample for electron microscopy, then you can do EDS on it. Like all electron microscopy, you need to prepare your sample in a way that it answers the questions you are trying to address. Erin will be discussing sample preparation in more detail later as it relates to their research question. I have a few images on this slide of samples I have successfully collected data with. Sections produced from frozen hydrated samples or traditionally prepared fixed and resin-embedded samples can be imaged on grids in both TEM and SEM, and when mounted on different substrates, such as for serial section array tomography SEM techniques. Small samples, such as pollen grains and cells, can be dried and mounted onto stubs for EDS, although it is worth bearing in mind that because these samples are not flat, you may get differential absorption of X-rays from the sides facing away from the detector. Samples embedded in resin and trimmed or frozen samples that have been planed to have a flat face are also excellent samples for EDS analysis. It should be noted that there is potential for element extraction at all stages of biological sample fixation, a schematic of which can be seen on the left here, with all the different pathways that can be taken, and I'm sure there are some techniques that have not been incorporated. The longer the fixation and sample preparation procedure is, the greater the extraction of ions. For this reason, rapid preparation techniques are better, including cryofixation and imaging frozen hydrated samples, microwave-enhanced fixation to speed up the penetration of chemicals into the sample, and using rapid embedding techniques, details of which can be found online. It is important that some form of sample preparation is completed to ensure that your sample is stable and conductive in the electron microscope, and when exposed to the electron beam. As mentioned, a relatively flat sample is required to avoid artifacts in data caused by absorption of X-rays, and you must ensure that your sample preparation does not extract your elements of interest. Often with biological electron microscopy, there is an optimization period where you need to test different sample preparation regimes, and it may be that your final results will require the use of more than one technique. Here we have an example that we produced in collaboration with researchers listed at the bottom of this slide, looking at nanoparticles in human tissue and in cell culture. The human tissue was prepared for routine TEM imaging using chemical fixation, dehydration, and embedding in epoxy resin. The samples were sectioned and placed onto a grid. The images on the left and the middle show an SEM image of the sample, and you can see a box on that image which is the zoomed in regions shown in the elemental maps. You can see that we are detecting titanium, chromium and cobalt nanoparticles within this structure, which is a lysosome inside a cell. On the right we have an image of macrophages that have been grown on plastic discs and fed cobalt nanoparticles. We used a higher kV on these samples to ensure the beam penetrated into the sample to detect the particles, which can clearly be seen as bright green clusters, and this required the use of the Ultim 170 detector. The SEM preparation for this involved fixation, dehydration, critical point drying and coating in carbon. In these results, we were able to localize nanoparticles in both human tissue and in cell culture. They also show that it is possible to get good research data from routinely fixed prepared biological electron microscopy specimens, and it may not be necessary to do a special fixation regime for EDS. It is also possible to look at unconventional samples, those without any stain. These are SEM samples prepared by fixing some leaf tissue, dehydrating and embedding the tissue in resin without using any stain for contrast. The sample was sectioned and placed into aluminium stubs. It was then imaged at 2.5 kV using our Ultim Extreme windowless detector. You can see on the images to the left that the nitrogen map in particular is very strong. On the right image we can see chloroplasts from leaf tissue. We can see that ultrastructural imaging using EDS maps is possible. These samples were not coated, but they were stable under low kV imaging conditions, which is only possible using the Ultim Extreme detector.
It is also possible to image cryosamples, and these are samples that have been frozen, are fully hydrated, and are being kept at very low temperatures while imaged in the microscope. These samples tend to be beam sensitive and can be challenging to image. Here we have a sample of plunge frozen freeze fractured wheat seed. This sample is courtesy of Jean Devonshire from Rothamsted Research. And this was imaged in a cryo-SEM at 15 kV using an XMAX 150, which is one of our large area detectors. The large area detectors we have are very fast and sensitive, and thus we're able to collect this very nice looking data within a single frame. You can see in particular the phosphorus, magnesium and potassium signals in the cells located in a line across the centre of the image, and these maps are particularly nice of this difficult sample. Here we have another example of a cryo sample, and this was also collected using an XMAX 150. And this sample shows an Arabidopsis thaliana trichome, which is a hair that projects from the surface of the leaf. These are very complex three-dimensional structures, and they easily get destroyed during traditional EM fixation and preparation. So in this example, the sample was frozen, and it was imaged in its frozen hydrated state. It was also coated with gold to help with conductivity. And you can see in these samples the distinctive distribution of calcium, silicone, manganese and phosphorus. As before, this data was collected at 15 kV, and in this case the elemental maps are an average of 2 frames. And now I'm going to hand you over to Erin, who's going to talk to you about her work in EDS on heavy metal stained resin embedded biological samples. Over to you, Erin.
Thanks, Louise. So you've just heard about the many sample preparation options available for doing EDS on biological samples, and in this section of the webinar I'm going to give you examples of applying EDS to heavy metal stained resin embedded samples. We've had the Oxford Instruments Ultim Extreme detector on our SEM in the Dunn School EM facility for about 5 months now, and have been using it with a range of different samples, including whole mount cells prepared for SEM and with resin embedded TEM samples. Some of these resin embedded samples have been specifically prepared for EDS, while others are archive samples from completed TEM or 3-view projects where performing EDS could give new and useful information about them. X-ray microanalysis is most commonly used with unstained samples, So we wanted to see what sort of elemental data we could obtain from some of our heavy metal stained samples and compare these to other types of preps. So in this webinar, I will discuss a few of our EDS projects, detailing how the samples were prepared and what sort of elemental data we obtained, with a particular focus on phosphorus. I will also give an example where heavy metal staining with osmium can actually provide useful information about the sample itself. First, I'll give you a quick overview of the setup we're using. I'll go into detail about the sample prep later on, but for most projects we're collecting 90 to 200 nanometer ultra-thin resin sections onto formvar coated copper grids, and we're not post-staining or coating them prior to analysis. The grids are loaded into the stem holder, which can take up to 12 grids at a time, and the secondary electron signal is obtained using the in-lens detector. We're using between 7 to 10 kV accelerating voltage, as beam reactivity of the sections is more of a problem at kVs below this. The ultimate extreme detector is optimized for working at or below 7 kV, so for the 10 kV scans, we're accepting a small increase in noise in our maps and spectra due to stray backscattered electron signals. However, we are in the process of testing different substrates for the sections to reduce charging so we can use lower kVs. The electron dose also contributes to charging. For most scans, we're going with a high dose to increase the X-ray signal, aiming to collect around 100 million X-rays per run, with run times ranging from 30 minutes to 2 hours. So I'll start with our erythroblast project, where we're studying a form of anemia called CDA type 1. CDA1 erythroblasts exhibit several abnormalities, including a particularly striking nuclear phenotype at the TEM level, shown here on the right and referred to as spongy or Swiss cheese nuclei. We're hoping that EDS will help us determine what these holes or inclusions in the heterochromatin are composed of, and whether they contain euchromatin, which will be high in phosphorus, protein, high in nitrogen, or something else. The erythroblast samples that we've done most of our analysis on so far were prepared using a standard TEM prep, outlined here on the left, with secondary fixation using 1% osmium and an overnight en-bloc staining with 0.5% uranyl acetate, followed by ethanol dehydration and infiltration with EPON resin. Animation. Here's a representative example of the elemental maps from these sections. There are strong signals from both phosphorus and nitrogen and some small qualitative differences between them. Since all of these analyses are spectral maps where X-ray spectra are collected at every pixel, post-analysis selection of elements and quantification of spectra from regions of interest can be done. This graph shows the elemental composition in different areas of a CDA1 nucleus. Importantly, lower density or lighter holes and the higher density darker holes contain comparable amounts of nitrogen and uranium to heterochromatin, indicating that they are likely to contain DNA. The phosphorus level is slightly higher in the heterochromatin, and we're analysing spectra from more CDA1 cells to quantify this further. If you're wondering about the aluminium and copper signals here, these are coming from the stem holder and grid respectively, and don't present a problem for this particular project because they're not what we're trying to detect in the sample. One thing worth noting when mapping phosphorus in osmium stained samples is that the M emission line of osmium and the K line of phosphorus are very close to each other. So to confirm that the phosphorus detected in CDA1 nuclei is not bleed through from the osmium, EDS was also performed on samples where osmium was emitted from the processing. In this case, the cells were cryofixed by high pressure freezing and freeze substituted with 0.2% UA and 5% water in acetone, then infiltrated with HM20 acrylic resin and polymerized with UV light at -45 degrees. The strength of the phosphorus signal in these CDA1 nuclei is comparable to that of osmium-stained cells, confirming that we are indeed detecting phosphorus and not overlapping osmium in those samples. This next project is also focused on mapping phosphorus. Arbuscular mycorrhizal fungi accumulate polyphosphate granules in their vacuoles. Rice root cells colonized by AM fungi contain electron-dense bodies in their vacuoles that closely resemble these granules and could therefore contain phosphorus derived from the colonizing fungus. We're working with Ronelle Roth from Cambridge University to determine if these structures in plant vacuoles do contain phosphorus using EDS. We're trying a few different sample preparation approaches for this, and I'll show you two of them today. The first is a normal TEM prep, but where the staining steps have been completely omitted. As expected, the contrast was quite low, and these sections were fairly beam reactive, which resulted in some distortion in the elemental maps over the acquisition. Phosphorus was detected in the fungus, but was not found in any of the presumptive phosphorus granules in the plant vacuoles. With this next prep, roots were cryofixed by HPF and free substituted with 0.3% uranyl acetate in acetone, then infiltrated with acrylic resin and polymerized with UV light at -50 degrees. Free substituting water with solvents at sub-zero temperatures is a gentler process than dehydration at room temperature or on ice, and should result in less extraction of elemental components than a standard prep. A disadvantage of this particular prep is that acrylic resins tend to be a bit more beam reactive than epoxy resins, which made longer data collections at high dose more difficult, and we will try EPON with this in future. We could also try cutting thicker sections with the acrylic resins. The fungus is outlined here in red, and large phosphorus-rich areas are detected throughout. Electron-dense bodies are present in the plant cell and contain nitrogen, oxygen and uranium, but not phosphorus. So we're detecting phosphorus in these samples, but are we detecting all forms of it? We're not detecting it in the plants. If the granules do contain polyphosphate, could this be more susceptible to the sample preparation procedure than other forms of phosphorus? Or could the phosphorus be washed out in the water bath of the diamond knife? Previous work by others on Phyton in Arabidopsis seeds used resin sections that were cut on a dry knife to avoid dissolving mineral deposits in the water bath, so this is something else we will try in future. I'll finish up with an example of how introducing heavy metals into your sample during the preparation procedure can have useful applications with EDS. Apex and Apex 2 are small peroxidases which are used as EM-compatible genetic tags. When treated with DAB in the presence of hydrogen peroxide, a highly osmophilic reaction product is formed, which stains therefore very strongly with osmium and allows the fusion protein to be localized at the EM level. No antibodies. Here is an example on the right of a previous Apex project we had in the EM facility, where a protein that localizes to both the nucleus and mitochondria was tagged with Apex. You can see that these organelles are much more electron dense in the cell expressing the Apex fusion compared to the non-transfected cell at the top. However, transfection rates can often be very low depending on the cell line, and Apex-induced staining may be more subtle in EM images if localized to different organelles or specific membrane sites, for example, on certain faces of the Golgi. So to save time at the TEM, samples can be pre-screened in the SEM using EDS. Here is an SEM image from the same Apex project, showing an Apex transfected cell on the left and an untransfected cell on the right. In this case, the contrast is very strong in the secondary electron image, but for different fusion proteins it might not be as obvious, and this is where looking at the elemental maps would be useful. You can see here that the osmium and oxygen signals in particular are very strong in the mitochondria and nucleus of the transfected cell. In summary, it is possible to extract useful elemental data from heavy metal stained and resin embedded biological samples, but there are many factors which influence our ability to detect certain elements with EDS. For instance, the cellular compartment it's localized to and how it's bound within that compartment affects how susceptible it is to being extracted during the dehydration steps, even during free substitution, as does its solubility in water. So it really is a case of just giving it a go and seeing what you get. If you can try a few different sample preparation approaches in parallel, for example, chemical versus cryofixation, heavy metal stained versus unstained, then that's even better. And heavy metal stains can also be useful for EDS, with osmium in particular having some interesting applications. For example, screening of apex staining is shown here, and also quantifying lipid accumulation, which is another project we're working on in macrophages at the moment. Louise has also produced an application brief with some other examples of differential staining in biological tissue, which can be downloaded from the Oxford Instruments website. And now I'm going to pass you back over to her.
Thank you, Erin. I hope you've been able to see that there are a wide range of applications for EDS in the life sciences, including cryo and room temperature samples, and in particular picking up on both the ultrastructure and composition of biological material. We've also shown a little bit of information about the interaction of biological samples with materials such as nanoparticles and pollutants. We've also demonstrated some low kV and low dose imaging solutions and shown that not only can we achieve sample stability, but also cryo-EM and cryo-EDS of frozen hydrated samples. And we've been able to utilise large area detectors and windowless detector technology for the rapid analysis of our samples. So hopefully you've seen that EDS technology has advanced now to the extent where imaging biological samples is beneficial and also provides us unique biological information. I look forward to answering your questions during the question and answer session to follow, and I want to reiterate my thanks to Erin for her contribution to this webinar. Thank you for watching.
Thank you for your presentations, Louise and Erin. It's now time for the Q&A. To ask the speakers a question, just type it in where it says type your question here and then press submit. So our first question, this one is for you, Louise, and it asks, what was the advantage of using the Ultim Extreme Detector on biological samples?
Thank you for that. The Ultim Extreme Detector has two main advantages that I can see. So first off, it is able to detect much lighter elements. In particular, nitrogen really comes clearly with the Extreme Detector, as I think you'll have seen on one of my earlier slides. The second one is that you can use a much lower voltage from your electron beam. Because it's windowless, you're able to then pick up and detect elements that are X-rays, that are produced using a lower voltage beam. The lower voltage beam reduces the specimen beam interaction volume, therefore you get better resolution, and you're able to detect more X-rays with a windowless system. So the Extreme works on two fronts.
Excellent. Thanks, Louise. And our next question asks, to what extent do you think sample preparation affects elements of different weights? So, for example, do heavy elements always stay in place better than lighter ones?
I can answer that. It's Erin. The answer, well, I can and I can't answer it. We're not really sure. But I think it depends a lot on how the elements are immobilized in the sample. So if they're free ions, say like calcium in the cytoplasm, it's possible that that's more likely to be extracted during the sample preparation, as opposed to if they're bound, say in DNA or proteins or within a membrane, like phosphorus and nitrogen in the nucleus. They're less likely to be affected by the preparation. So I'm not sure if it depends on the weight of the element as much as where it is and how it's immobilized in the sample.
Excellent. Thanks, Erin. And our next question is also for you. as well. And this one asks, why would you use EDS on stained and embedded samples? Aren't these elements removed during the fixation process?
So yeah, this follows on a bit from what I was saying just then. Is it really depends on where the elements are in the sample, because there will be some effects of the sample preparation process with a standard TEM process. Probably the biggest culprit would be the dehydration step, where things can be lost during that process. And there are things you can do to mitigate that, like perform the dehydration and the resin infiltration at lower temperatures, for instance. But I think there's lots of good reasons why you would do EDS on stained and embedded samples. We've seen several examples in this presentation where you can get very nice elemental maps from samples that have been prepared for standard TEM. So it opens up the door to doing X-ray microanalysis on samples that have already been prepared, so you can get more information from any blocks that you might have that have previously been imaged with TEM. And also because a standard prep is fairly routine, it also makes it an accessible technique. And yeah, as I said, it's compatible with TEM as well, so you can get the maximum amount of information, hopefully, from your sample.
Excellent. Thanks, Erin. And our next question is for you, Louise, and this one asks, what challenges do biological samples pose for EDS?
Thank you. A lot of the challenges are similar to the challenges biological samples pose for EM in general, in that they are non-conductive. So especially for SEM, you may need to have a coating on the sample. They're also composed of light elements, so not only do they not scatter electrons very well, they don't produce as many X-rays as heavier element samples. So for those reasons, you do need particularly sensitive detectors. The other challenge is biological samples are not flat. They've got very 3D structure. So during the sample preparation process, you need to make sure that we have a relatively flat surface. And I gave a few examples, such as sections prepared on grids, or if you've got a resin block, you can plane the surface of the block to make sure that you've got a flat surface. And the reason the flat surface is important is because you can get selective absorption of different X-rays, and you don't want to skew your data as a result.
Excellent. Thanks, Louise. Our next question asks, with thicker sections, how thick can one go? Do you lose any spatial resolution from the electron beam interaction volume? Louise, is that something you can help answer?
Yeah, perfectly happy to answer that. I think the question depends whether it's TEM or SEM based. So TEM, yes, you will lose resolution with thicker samples. With an SEM, the resolution depends on the electron beam interaction volume. So you can have very thick sections but still only sample a relatively thin layer at the surface of the sample with a low energy beam. So with that, with the SEM, it's the microscope conditions that are going to play more of a role in your resolution than the thickness of the section.
Excellent. Thanks, Louise. Our next question was wondering if you can just confirm what the optimal working distance for the extreme was.
The optimal working distance is generally set when the extreme detector is installed. So each microscope configuration is going to have a slightly different recommended working distance and optimal working distance. It's usually listed on Aztec in the software, so your engineer would actually
put it into the software and it will be shown there. And the working distance cited there is the working distance from the sample to the pole piece of the lens, of the objective lens. On the systems I've looked at, it's been around 10 millimetres, which isn't the same as the working distance between the sample and the extreme detector, as that sits just slightly below the pole piece, so that's a smaller working distance.
Excellent. Thanks, Louise. Next question is for you, Erin. And this one asks, what type of biological sample preparation would you recommend for EDS?
I think Louise covered this quite well in her presentation. It depends on your question, and if you're not sure, I think which approach to take, I think the best thing would be to do a standard TEM sample prep where you could include all your heavy metal stainings and maybe in parallel do one without heavy metals, and then that would be a good starting point and you can see how you go from there.
Excellent. Thanks, Erin. Next question asks, is it possible to fix cells over gold-coated glass slides for EDX and SEM analysis?
Yeah, I'll answer this one. Yes, it is possible. I've done, I think I showed you in the presentation, some macrophages, and they were on a cover slip, and that didn't have any special coating on, but there wouldn't be a problem incorporating a gold coating into that to help the cells adhere to it. Plus, it would have the advantage of making the substrate much more conductive. So the answer to the question is yes, it is possible.
Excellent. Thanks, Louise. And our next question asks, what is the difference in using TEM or SEM for EDS on biological samples?
So there are a few differences in our detectors and the way that we operate the microscopes for TEM and SEM. In a TEM, we'd have to operate it in STEM mode so that we can scan across the sample. And in general, the sample actually has to be tilted slightly towards the detector in order to get the best angle for producing X-rays. In TEM, you've got a very high main beam energy, so you actually activate all of your X-ray lines that may be of interest. And in SEM, because you've got a lower KV setting and you've got, it depends what detectors you've got, but for example, if you're using extreme and biological samples and you wanted to use 5 kV, you may have to look at what elements you're trying to detect, because some of the high energy X-ray lines might not be activated by your beam energy. So there's a little bit of difference with regard to microscope conditions between the two techniques. With regard to biological sample prep, I hope we've been able to say a few times that you can do TEM sample prep for an SEM and you're able to collect EDS on it without an issue.
Excellent. Thanks, Louise. And just a quick reminder to our live audience, you still have time to ask a question. All you need to do is just type it in where it says type your question here and then press submit. So our next question asks, why is the element hydrogen not detected through EDS? Louise, is that something you can help answer?
Yes, I can answer that question. So hydrogen doesn't produce a lot of X-rays. It's quite a rare occurrence. And the X-rays that are produced by hydrogen are such low energy that it is very, very difficult to actually detect them. They'll most likely be absorbed by the materials made to construct the detector. So for that reason, hydrogen can't be detected with current technology.
Excellent. Thanks, Louise. And our next question is for you, Erin. And this one asks, what EM labels could be used in combination with EDS?
Yeah, so you can use quite a few different labels. So you can introduce nanoparticles into your sample via secondary antibodies, which are conjugated to colloidal gold or nanoprobes of different elements. So you can have gold and silver in the same sample, and which can be quite difficult to differentiate between different particle size if you're using quite small probes at the TEM level, but if you go to EDS you can differentiate based on their composition. What else? So there are different tags. So I spoke about Apex in my presentation. That's a genetically encoded tag for EM. There's also other ones like Minisog, which works in the same way, where you get localized accumulation of osmium, which tells you where your fusion protein is. So screening with EDS could help localize cells that are expressing that. There's also some other tags, so ferritin tags, and also different heavy metal stains which may differentially stain different organelles and components of your sample, like osmium and lipids.
Excellent. Thanks, Erin. Our next question, I think, is something that you've kind of touched upon briefly before, but it asks, is it possible to use a sample prepared for TEM or SEM for benchtop microscopic X-ray fluorescence spectroscopy analysis? Louise, is that something you can help answer?
Yes, it's possible to use a sample prepared for TEM or SEM on benchtop microscopes for EDS. So the answer is it is possible. You may have to look at what resolution you can achieve and what information you need out of the sample, but there shouldn't be a reason why you couldn't do it.
Excellent. Thanks, Louise. Next question asks, do you have any suggestions for making a 3D EDS analysis of biomass materials successful at room temperature? Can either of you help with that question?
Yeah, I think I can. So it does depend on the materials. I'm not entirely sure which materials are being referred to, but there are a variety of options to do 3D EDS. In particular, I've been doing some work using array tomography, in which we take serial sections that are collected on an ultramicrotome outside of the microscope, and they're put on a substrate. And then we can go and image every single section sequentially in the microscope, and in that way we can build up three-dimensional data. And I have done this with EDS quite successfully. So at the moment it's a very manual process because there's no automated way to do it. But it is entirely possible to do it, and you can build up some very nice maps in a relatively short amount of time.
Excellent. Thanks, Louise. Our next question asks, if one is using a windowless detector, how does one protect the detector itself from surface contamination? Louise?
Oh, I can answer this one from a user perspective. So basically you have to make sure that whenever the detector is cooled, the microscope's under vacuum. So after you are finished using the detector, you warm it up, retract it and warm it up, and then you need to wait a good 20 minutes to make sure that it's properly warm before you vent the microscope. So that's very important. And once it's warm, then it's okay to vent the microscope and you don't get contamination.
Excellent. Thanks, Erin. Next question asks, is it possible to define the colour chemistry in a cell?
Hi, yes. I'm not entirely sure what the question is asking in this case, but I'm going to take a guess as to what it is. So, are we able to define the chemistry in the cell using colour imaging or colour EM? I'm thinking that's what the question is asking. And yes, EDS is a very good way of conducting colour EM and using the elements and the chemistry within the sample itself to provide compositional data that can be displayed as a colour map, and in that way it can give additional ultrastructural information over the normal grayscale electron images that you would achieve.
Excellent. Thanks, Louise. Our next question asks, do you think there are any advantages of performing, for example, cryofib lift over sectioning? Erin, is that something you can help answer?
Yeah, well, my first thought is that cryofibs are pretty rare and it's quite difficult to do cryofib lift outs. So from that side of things, I'd say it's more technically, much more technically demanding. But if you do the cryo lift out, then let me think, you get less artifacts than you do with, say, cryosectioning. So with cryosectioning, you do get sort of compression artifacts, whereas you don't have that with cryofib lift out. So that would be one advantage.
Excellent. Thanks, Erin. And our next question is also for you. And this one asks, what advantage is there in using EDS combined with APEX?
So this, I think, is something I'd like to explore more. And the example that I showed in the webinar was one where the APEX labelling was very strong. The osmium signal from the APEX reaction was quite strong. I think if you have a much lower density of osmium staining from APEX, using EDS could help you to screen the sample to find cells that may be expressing APEX, particularly if you have a low transfection rate. We had a project a while ago which was similar to this, and it was very difficult to find cells that were expressing APEX. So this could be, using EDS, could be one way that you could help screen the samples and narrow down the time that you spend on the TEM with your APEX imaging.
Excellent. Thanks, Erin. Our next question is for you, Louise, and this one asks, how were the wear particle TEM samples prepared for EDS in an SEM?
Thank you. Yes, they were prepared relatively routinely. So these were samples from patients. So we had no control over the sample preparation with regard to the initial fixation. So the initial fixation was an aldehyde fixation, and they were then rinsed and embedded in a resin, and it was an epoxy resin, and then sent to us. So they didn't actually have a lot of heavy metal staining in the fixation process. And for the SEM, they were just put on TEM grids. They were actually imaged in a TEM quite extensively prior to being put in the SEM, and so the SEM sample preparation was relatively easy. And there are a couple of ways you can do it. You can either mount the TEM grids directly onto aluminium stubs and using carbon sticky tabs, or you can get small stubs that are designed to fit TEM grids in, and in this case I used one of those. So it was just a little dip in the stub itself, and the grid just sat within the dip, and then I was able to collect the data in the SEM. So beyond normal TEM sample prep, there was nothing special that needed to be done.
Excellent. Thanks, Louise. Our next question asks, what should be the ideal probe current range of a microscope to get a good biological EDS data?
I think it depends on your sample, what how abundant your element in there is that you're trying to find, but also how you've prepared your sample affects how beam reactive it is. And so if you're working with higher probe currents, you might get more X-ray signals, but you can have more sample damage as a result, and so it will be moving a bit more as you collect your spectra, which will affect the spatial resolution. So with the work that we've done, I try to work at fairly high currents to maximise the X-ray signal, but sometimes you can't. It depends also on the resin that you're using. Some resins are more reactive than others, so you have to adjust the kV, the dose, and your scan times as well to mitigate or to find the optimal settings for your sample.
Excellent. Thanks, Erin. And our next question asks, what is the largest solid angle achieved using these detectors in the SEM and or TEM?
The largest solid angle for the TEM is 2 steradians, and for the SEM is about half a steradian with these detectors.
Excellent. Thanks, Louise. Our next question says, many modern SEMs routinely use partial pressure in the sample chamber to permit examination of non-conducting materials. How can or how will a windowless detector function in such instruments?
It's a good question, and thank you for it. I'm aware that many SEMs, particularly those that are doing 3D SEM, use variable pressure or environmental pressure settings within the SEM to help with charging samples. However, for our windowless detector, it's not advised to use it in those conditions. The windowless detector does require a high vacuum. So if the instrument being used needs variable pressure quite regularly to image these samples, would actually advise using one of our window detectors such as the Ultim 170, which still has sensitivity and extremely fast, but the window will protect the detector.
Excellent. Thanks, Louise. Well, I'm afraid that is all we have time for today. I would now like to thank Dr. Louise Hughes and Dr. Erin Johnson for their presentations and for answering your questions. I would also like to thank the webcast sponsors, Oxford Instruments, and of course you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand in nature.com webcasts. Thanks for watching, and I hope you'll join us again soon.